our purines, what is your answer? Give me your answer in the text. What are the two rings that are present in our purines? You have to text me the answer, okay? I told you yesterday that purine is a heterocyclic structure. No, it's not benzene, Vijendra. Okay, it's not benzene. So, anybody else? Do we have any other answers? Okay. Why do we call purines as purines? And you know pyrimidines? Okay. Vijendra, I will tell you something. If you see audio is not proper. Okay, give me. Can you hear me well, students? Now is it clear? That's why I gave you a technical question. Is the audio, yeah, clear, right? So I was asking you the simple question. Okay, Vijendra, I was talking to you. So the thing is, when you told benzene ring, I really understand that you went for that six atom structure and you called it as a benzene. So my question is purine. So if you look at the picture that presents purine, it's not just one ring. There are two rings there. So I'm just asking my question again. What are the two rings that are present in our structure of our purines? Okay, what is the names of this? If you understand, okay. Come on, students. Okay. Okay, it's all right if you're wrong. We can just keep on correcting. Exactly. Okay. Imidazole is the answer. So I can check the chat box. Okay. Imidazole. Somebody said, let me check. Let me tell. Jaydeep. Excellent. Okay. So he says it is imidazole ring and it is perfect answer. So tell me, what made you call that it is an imidazole ring? Can you tell me what are the atoms that are present in this particular ring? Three hydrogen. Not really. Three nitrogen. That's right. Three nitrogen add. I think some of you are sitting in a classroom, right? Some say four carbon. All right. Okay. Okay. Yes. It's nice that you are participating. And Jaydeep, you're right that it is three nitrogen, and the remaining two atoms are carbon atoms. Okay. So three nitrogen, two carbon atoms. Ring is called as imidazole ring. And coming back to Vijay, I think. Vijay, when you told that it is benzene ring, it's actually not benzene ring. So if you can see this cytosine, thymine, and uracil structure, it is made up of six carbon atoms, isn't it? So these six atoms, I'll tell you, six atoms. So this six atom ring is called as a pyrimidine ring. The reason why we call pyrimidine ring is because if you just check the structure, it is made up of around four nitrogen atoms and two carbon atoms. So this four nitrogen atom and two carbon atom structure is called as a pyrimidine ring. Whereas in purines, it is made up of both your pyrimidine ring as well as imidazole ring. That is why we call our purines as heterocyclic structures. Is it clear? Right? So you just have to remember, and it's very easy. Okay, it's not too complicated. I know that you people are very smart to remember things. Anyways, oh, we talked about our nitrogenous bases, and we talked about what are purines and what are pyrimidines and what are their structures, isn't it? So now we have to, go for the next ones. Let's see. So when we talked about what are nucleosides, and we really talked about what are the sugar uh, carbon atoms that are involved in the formation of a nucleoside. So can anybody tell me which carbon atom does a nitrogenous base attach to our deoxyribose sugar? Should I repeat the question? Yes, carbon atom 1. Perfect. So to the first carbon atom, our nitrogenous base comes and attached to the deoxyribose sugar, and that will make the structure as what? We have deoxyadenosine, deoxygynosine, deoxycytosine, and then we have deoxythymidine. So these are the structures, right? So nucleoside, okay, nucleoside. Anyways, One second. Yeah, it's already recording, no problem. Yeah. So now when we have to go for the next concept, so we understood what are nucleosides, and then we are talking about nucleotides. So in nucleotides, again, it's very simple for you right now, because it's very easy for you. It should be very easy for you to tell this answer. Which of the carbon atom that is involved in the formation of a bond between a sugar group and a phosphate group. So immediately it should be 5' or the fifth carbon atom. So once we have this sugar, and then once we have this nitrogenous base, and then we have our phosphate group, so that will make us nucleotide. So the nucleotide is the basic monomer of our DNA. Okay, so you know what is a monomer, right? It is the simplest unit of a particular structure, so that is called as a monomer. So here we have a nucleotide, and the nucleotide names goes like this chemically, that is deoxyadenosine monophosphate, deoxygynosine monophosphate, deoxycytidine monophosphate, and deoxythymidine monophosphate. So these are the names for nucleosides. So next topic that we understood was how is the formation of a polynucleotide strand takes place. So the formation of a polynucleotide strand takes place between the two adjacent nucleosides. So when we talk about two adjacent nucleosides, that means one nucleoside below the other nucleoside. We are not talking about the opposite nucleosides. Yesterday, I was emphasizing on this so much. I was telling you, if you are asked to write the structure of a DNA, and if you just assume that this is the structure of the DNA, when you're talking about a polynucleotide chain, you're literally talking about this one particular chain, not this one. Okay, if you're talking about the other chain, then it is this one. Okay, so this one chain is called as a polynucleotide chain, and even this is called as a polynucleotide chain. So if you're talking about the formation of a polynucleotide chain, you are talking literally about how this line forms. So that means you're talking about how one nucleotide is attached to another nucleotide. So when we were talking about how one nucleotide is attached to another nucleotide, so let us get back to our basics. So we just talked about there are 5 carbon atoms in our deoxyribose sugar. In that 5 carbon atom, we just try to give importance to the 5th carbon atom and the 3rd carbon atom. So now what happens is, in our 3rd carbon atom here, this is a 3rd carbon atom, there is a free OH group. Okay, there is a what? There is a free OH group. So this free OH group will participate with the phosphate group of the another. See, this is the phosphate group, and this participates with the OH group of the next next nucleotide. So this phosphate group and the OH group, once they come together, they form this bond called as phosphodiester bond. Once they form this phosphodiester bond, now a water molecule goes out, thereby forming this covalent bond that the phosphodiester bond forms. Now this will create a polynucleotide chain. Now there is one small question here, so let me ask that question and see what is your answer for that. Okay, my question is very simple, students. So my question is, just listen to the question properly. When a phosphate group, okay, when a phosphate group attaches to the 5th carbon atom of your deoxyribose sugar, I'll repeat, when a phosphate group attaches to the 5th carbon atom of your deoxyribose sugar, what kind of bond formation takes place? So this is my question. Let me find the best answer. Yes, phosphoester bond. I'm very happy. Why? Because you guys did not tell phosphodiester. You see, there is a lot of difference between a diester and an ester. Okay, so the answer is right. Somebody, I don't know if you just typed it in a hurry, you just wrote ester bond, so you just add phosphoester. So that will be the perfect answer, right? So giving the right answer. So the answer is phosphoester bond, and you're absolutely right, kids. Okay, so let's get back to where we left off. So we understood the the formation of a polynucleotide chain. So once we have the polynucleotide chain, we understand that there is a bond between the 5th carbon atom and the 3rd carbon atom here, where the water molecule goes out, and then we have a phosphodiester bond that will create the chain of our polynucleotide. So once we have our polynucleotide chain, the next thing what happens is the two strands come together, they form a helical structure, and between these two strands there are hydrogen bonds between the adjacent, the opposite nucleotides, which are complementary to each other, and that is when you have your both helical structure of your DNA. And yesterday I had given you an assignment, and I had asked you to find out who was that scientist who did the first X-ray crystallography studies of our DNA. So do you have an answer for me today? Which scientist performed the first X-ray crystallography studies of DNA? Did you search, or did you forget, or didn't you get? Just a second, students, okay. Which scientist performed the X-ray crystallography studies on the DNA that was used by our Watson and Crick to propose the double helical structure of DNA? Okay, who did that? All right, I'll give you some time to think about it, and let's see if you can come up with some answers. It's a very, very important contribution. That's why I asked you guys to find out, because you just go with Watson and Crick, and it's very, very easy to remember these names because they're Nobel laureates and they've been their name has been emphasized for such a long time in the history of science. So, anyways, so coming to the proposed model of the Watson and Crick, so now you guys have to remember that it should be in your heads that already the structure that we are going to study in the double helical structure is the B form of DNA, or the B DNA structure. And what kind of a helix it is? It is a right-handed helical structure. So Watson and Crick, when they proposed, the first thing they told that the DNA structure is a right-handed helical structure, okay, and it goes like a staircase, like a structure. So if you look at the structure of the DNA, now what happens is you can see that the sticks, right? I will just put it here, okay? The sticks of the DNA, that is the backbone of the DNA, you can call. So these like ladder-like structures, or the backbone of the DNA, is made up of two important compounds. One is the sugar and the phosphate. So we just call it a sugar phosphate backbone, and the sugar phosphate forms the backbone of your what do you tell? Your DNA, whereas the nitrogenous bases, they project towards inside where they form the hydrogen bond. Now, the DNA structure is double helical, so there are two polynucleotide chain which are involved in the structure of the DNA. One chain has the polarity from 5' to 3' direction. So now we should know that the 5' represents the free phosphate end and the 3' represents the free OH end, right? And similarly, the opposite strand is anti-parallel. So if one head is towards the phosphate end or the 5' end, the opposite you can see here. If one chain is running from fifth, the other chain would be running from third. So this shows the anti-parallel nature of our DNA. And the third thing is we talked about the base complementation, that is the complementary nature of our DNA. So when we talk about the complementary nature of the DNA, so we are quite aware that they have the base complementation. That means the adenine always complements with thymine with 2 hydrogen bonds, and the cytosine complements with gynosine with 3 hydrogen bonds. So this is called as base complementation. And yesterday we even worked out an example of a DNA strand. So how we can complement, so and how we can predict if we have one chain, and we can easily predict the other chain, correct? And next one is the configuration, the physical configuration of the DNA. That is, if you look up and then see the diameter of the DNA, that means it has only 20-degree angstroms of the diameter. And then if you see the turns of our DNA, so each turn is around 34-degree angstroms, or 3.4 nanometer of length is there for each turn that the DNA takes. And between the two turns, there are around 10 base pairs. So since there are 10 base pairs, see students, what happens is, if you see molecular biology, if you see in the high level books, so they say that there are around 10 to 11 base pairs or 10 to 12 base pairs. But logically what happens is, on an average, there are only 10 base pairs that are present per turn of the DNA. So since we take 10 turns as a reference, if you take 10 base pairs per turn as a reference, that means per each base pair, there is around 3.4 degree angstroms of distance between each base pair that is stacked upon each other. And then if you convert that into nanometers, it is around 0.34 nanometers of distance between the two base pairs that we have in our DNA. And we talked about what is major groove and what is minor groove that we have in the helical structure of the DNA. So this was about the structure of DNA that we talked. And we also discussed about the other possible forms of DNA, that is aDNA and the zDNA. So there was a question, so which is the structure of DNA that we can extract using the bDNA in a laboratory condition? So this was a question that was asked some years ago. So the answer would be the aDNA, as we discussed. So what do we do? We take the bDNA, which is the right-handed helical DNA, which is found in 98% of the living organisms, and then we go to the lab and then we try to dehydrate it. So we know that how do we dehydrate it? We either use strong alcohol or acids. So now the dehydration reaction takes place. So when the dehydration reaction takes place, we remove a lot of water molecules in the structure of our DNA. When we take a lot of water molecules in the structure of our DNA, now what happens is the length is there, right? The groove, that is the 34-degree angstroms. So the number of base pairs in the groove will start increasing. And then now what? The compression of this structure takes place. When the compression of this structure takes place, now you can see that the structure is more condensed and the number of base pairs increase between these turns. And this is what is called as the A form of DNA, or the laboratory DNA, which has no clinical significance or biological significance for us to for our living organisms. Whereas when it comes to zDNA, it's extremely rare, and it has been very few times or some rarely spotted in plants. So the zDNA is quite longer DNA. That means for one turn, it is not just 10 base pairs. We can expect up to 12 to 14 base pairs which are stacked. Sorry, 8 base pairs which are stacked. I'm really sorry. So the distance between the two base pairs is quite large, and the turn is towards the left-hand side, not the right-hand side. So this is about the zDNA. And you remember one thing, which form of DNA is called as the laboratory DNA, that is your aDNA. Which form of DNA is the left-handed helical DNA, that is your zDNA. Very, very, very important points with respect to your examination point of view. All right. So we understand that the base complement each other in the DNA structure. When they complement each other, when adenine goes with thymine, it forms 2 hydrogen bonds, and when cytosine goes with guanine, it forms 3 hydrogen bonds that we discussed. And we also understood the importance of these hydrogen bonds in giving the extra stability for our DNA structure during packing or during the translation or transcription process. So next we discussed about Chergaff rules, how purine and pyrimidine ratio is equal to 1 with respect to our DNA, and which gives us the stacked nature or the perfect configuration to the structure of the DNA. So this was done. We'll talk about it furthermore, right? So here is where we left off when we're talking about the central dogma of life, and I was giving you just a simple explanation on what is central dogma of life, or what is central dogma. The pass of information or the flow of information that happens in all living organisms from the DNA to the protein level. So that is called a central dogma. And I was also telling you what is this flow of information and why we have to understand this flow of information. I told you, when you look at this living organism or the living system, it is such complex biological body. It has thousands and thousands of functions that it has to take place. It has to take care of. So when we have thousands of functions that it has to take care of, who is controlling all this, or who is telling our cellular organelles or cells to behave in such a way, produce such certain enzymes, produce certain proteins, produce certain hormones at a specific time, at a biological times? So how is it even possible, and who is responsible for that? So we credited all that to the messages that are present in our DNA in the form of genes. And we also discussed that not 100% of the DNA that we have is responsible in holding up the information. I told you that it's only rarely 2% of the DNA in a living form is functional, and the rest of the 98% is not at all functioning in any way. okay, which we call it as the junk DNA. We'll talk about it. So the functioning DNA, which we call it as a gene, so they pass on the messages that are hidden in these genes in the form of mRNA or messenger RNA or the readable messages for our cellular organelles to construct proteins. So when the RNA messages are read by our cell organelles, they construct proteins, and the proteins are the molecules of biological function. That's what I told you. 98% of your biological function is carried out by proteins, and we just discussed about it, right? So now we have to talk about where is this DNA present and how it is packed and why we have to study the packing. So let us understand by the lowest organism, that is the prokaryotic DNA. So when we talk about prokaryotes, so we already know that in our first year, we studied that prokaryotes are the unicellular organisms and they do not have, what do you tell, membrane bond organelles. So when they don't have membrane bond organelles, majorly they miss this nuclear membrane. When they don't have nuclear membrane, we call them prokaryotes, and the kingdom Monera comes under prokaryotes. That includes archaebacteria and eubacteria, right? So when we see that prokaryotes don't have a cell membrane, so that nuclear membrane, that makes their nucleus just to float naked in the sea of cytoplasm around them. So it's just floating. It's just a mosaic structure that is just floating in the cytoplasm. Now, this DNA is not scattered everywhere. It is just within this mosaic nucleus of our prokaryotes. Now, if you see, there are certain proteins that are involved in which this DNA is held together, and this entire region where the protein just holds up all this DNA together, so this structure is called as a nucleoid. So the entire nuclear structure of our prokaryotes, we call it as a nucleoid, right? So when we see the DNA that is present within the nucleoid, they are present as large chunks of DNA, which is not quite organized as chromosomes like that we see in the eukaryotes. So if you perform an onion root tip experiments, you can easily see that it is nice X-shaped bodies that the DNA is organized into in eukaryotic cell. But in prokaryotes, it's not like that. It is just like you take some part of the thread and you just wind up to make one loop, and then you have another chunk of threads, and then you just pack it up to make another loop. So it's similar to that, and it's just happening here and there, and that is how the DNA is organized within the nucleoid of our prokaryotes. So now, when we look at the prokaryotic DNA, so usually very less of this is spoken in your textbook about the prokaryotic packing. So understand that DNA has a negative character. That means it has a negative charge in it. So now this negative charge DNA requires a positive energy or the positive protein in which the attraction takes place between this positive and the negative bodies, and then the winding of the DNA takes place around them. So since the prokaryotes are very, very primitive organisms, they do not have a very sophisticated proteins like our eukaryotes. Instead, what they have is within this nucleoid region of our prokaryotes, they have the special proteins called as scaffold proteins. So the scaffold proteins, so you just have to remember this scaffold word, okay? So the scaffold proteins are non-histone proteins that are present in our prokaryotes. So now they do not have an organization like our eukaryotes. We'll just talk about it. They just come in few chunks. It can be bigger, it can be smaller, it can be 2 units, it can be 4 units. So the number of units, the amount of protein, or the position of the protein is not well defined in prokaryotes. So that makes them DNA to be just wound into small bunches and into a thick what do you call, like noodles in soup. Okay, so that's how the prokaryotic DNA is packed. Understood? So what we have to take from this is the prokaryotic DNA is packed by special proteins called as scaffold proteins. They lack nuclear membrane. The nuclear region is called as nucleoid, and the DNA present in them is called as nucleoid DNA. And this nucleoid DNA is packed by these scaffold proteins, and another name for the scaffold proteins is called as non-histone proteins. All right. So this is about prokaryotic packing, and usually there is a lot of mol bio with respect to how this DNA is packed with respect to prokaryotes, but to your level it is not so significant. So we'll just move on. Okay. So when it comes to eukaryotic packing, eukaryotes, as the name suggests, it is quite complex cells. So eukaryotes, in spite of having this, membrane bound organelles and very organized nuclear structures, so you can see the complexity and the way they are packed. All right. So let's see. In eukaryotes, what happens is we have specialized proteins, and these are called as histone proteins. As I have already mentioned, the histone proteins are positively, they have to be positively charged proteins. And for them to gain positive charge, students, it's very, very simple to analyze the biochemical nature of anything based on the ratio of what chemical compounds they are present. They have, correct, right? For example, if you have 20 people in your classroom, let us say that each of you is a particular type of amino acids. So if you don't know what are amino acids, then it is very bad of you. I don't tell bad of you. That means you have to rebrush your memory, going back to your biochemistry. Okay, you know that amino acids are the basic units of your proteins, right? So you have an amino acid. An amino acid can be positive, it can be negative, it can be neutral based on the type of charge it bears in its functional unit. All right. So we have positively charged amino acids, we have negatively charged amino acid, and we have neutral amino acid. Let us say that we have a protein like you are all proteins. Okay, let's say we are all proteins. So your classroom is a protein, and your classroom has 20 amino acids, that is 20 people of you. In that, 18 of you are positively charged and 2 of you are neutral or negatively charged. So what will be the net charge of your classroom? Positive, isn't it? So it will be more positive. Similarly, based on what type of amino acids are involved in the formation of these histone proteins, that gives them the net charge. In this case, the type of amino acids that are present in our histones are filled with this basic amino acids. Remember this, the basic, okay? They are chemically, they are basic amino acids, and the examples are lysine and arginine. So the lysine and the arginine amino acids are high in number in our histones. So since they have high in number of this basic amino acids, that gives them a very net powerful positive charge. Now, when we talk about histone can be one monomer. So a see, for a histone to be functional, it requires its friends. And this friends, we call it as a histone octamer. So I want to be participating in the packing of DNA. If I want to be participating in the packing of DNA, then I alone cannot finish the job. If I have to finish the job, then I need 7 more of my friends. So what are those friends? If you just look at your histone octamer group, you can see that there are specific type of histones. What are those? There are H1, I'll just H1. Here we have H2B, we have H2A, and then we have H4, and then we have H3. You get my point? What are the histone octamer proteins? H1, H2A, then we have H2B, then we have H3, and we have H4. So these H1, H2A, H2B, H3, H4, they are all the names of the monomers of the histone octamer. Did you understand? 1 units. So now what happens is they will come together. What will come together? Majorly, you can just observe this picture. H2B, H2A, then we have H3, and then we have H4. So they come and form this histone octamer. Understood? So they form this ball. So once they form this ball, now they're very positively charged ball. Now each ball can start winding up around 150 base pairs of DNA, which is negatively charged. Did you understand? So have you played this tap? In our Kannada, we call it as bugri. So tap, that means you have this wooden toy with a nail under it, and then you start winding the thread around it, and then when you release it, the trap starts spinning. So now let's not go to the spinning part. Let's go to the tap and the thread part. So you start winding the thread around this nail and the body of the tap. Similarly, you have a ball of your histone octamer, and the DNA is wound very, very, very tightly around 150 base pairs of DNA can be wound around one histone ball or one histone octamer. Now, what happens? If you have, I'll just see, okay, we have a picture. Yes, perfect. So here we have one histone ball, isn't it? And then here we have another histone ball. Now you can see that a small tail portion of the DNA is extending from one ball to another ball. Correct, right? So you can very clearly see that. If you just observe this picture, there is one more histone. So that is called as H1 histone. So usually the another name of this H1 histone is called as linker. Okay, we call it as linker histone. Linker. Okay. So what is the role of this histone linker or linker histone? Okay. Even now my pen tab is not functional, so that is such a sad thing. Okay, anyways, so I just write it as li liquor, so it's actually linker. I'll just write it as n students. Okay, so linker, so that is spelling. So I better get rid of that spelling as soon as possible. Anyways, so now the H1 histone is called as the linker histone, and the DNA that extends from the one histone octamer to the other histone octamer is called as the linker DNA. All right, did you understand this? Are you able to follow me? First of all, that is very, very important. Are you able to understand? Okay, just give me an emoji reaction faster. You have time. Perfect. Okay, nice. So if you guys have any doubts, please put it in the message. All right, so let's get back to where we left off. Okay, so chalo. So I was saying, so we have one histone ball and we have another histone ball, and both of this histone ball are connected by this linker DNA, and the histone that is supporting this linker DNA is called as H1 histone. Now what happened? We have 150 base pairs, here 150, see, here 150, here 150, here 150, like that the base pairs are being stocked, okay, very systematically. And then if we just observe it under the microscope, okay, if I just observe it under the microscope during this stage, it looks so pretty. So how does it look? It looks like the pearl necklace. Okay, it looks like exactly like a pearl necklace. How does a pearl necklace look? You have a string and you have the balls of pearls so systematically arranged, isn't it? That is exactly how your nucleosomes look like. So what did I just mention? So this pearls on the string or the beads on the string model is also called as a nucleosome. Understood? So first it was seen under the microscope, okay, and it was visible to the eyes, so that's why it was called as beads on the string model. Okay, so and it is also called as a nucleosome structure. Okay, so once we have this nucleosome structure, now what happens is the further condensation of the nucleosome takes place. So when does this usually happens? So the typically when you observe the structure of our DNA, now in most cases in our cellular cycles or in the cell functions, they're very loosely packed. Okay, they're very, very loosely packed, and they're not present as chromosomes, so they're mostly present as nucleosomes, okay, or chromatin fibers, you can tell. Either nucleosomes or chromatin fibers, that is how they are just present in the nucleus in most of the time. That means whenever the cell is metabolically active. But when it comes to the cell division, when it comes to that, okay, I have to divide mitotically or I have to divide meiotically. So at that point, what happens is a proper condensation process of this DNA takes place. And the reason is very simple. If you want to go on a trip, you just don't stuff everything in a bag and take, because you tend to miss something, or you tend to occupy more space in your bag, or the travel becomes very uneasy. Similarly, they are going to just another different daughter cells. So that is why what happens is this DNA has to be packed very, very systematically and uniformly. So that point, what happens is the further condensation of your nucleosomes takes place. And the nucleosome condensation takes place through chromatin proteins and non-histone proteins. So these chromatin proteins and the non-histone proteins, they come in between this linker DNA as well as the nucleosome DNA, and then they start winding and rewinding, making it more condensed and more thicker thread-like structure. And this thicker thread-like structure, we call them as chromatin fibers. So once we have this chromatin fibers, more chromatin proteins will come, and more of this chromatin proteins will start condensing the DNA into chromosome structure. Understood? So we have chromosomes now, which are very beautiful structure, which you can see during the metaphase of our cell division. So now what happens once we see this chromosomes? Now there is one small thing that we can observe. If I take an X-ray of the chromosome and if I observe the structure of the chromosome, what I can see is at certain regions of my chromosome, there are dark bands. Dark bands means they're like very dark colored. Whereas in certain regions of my chromosome, there are very light bands. So this shows something. What does it show? Wherever I have the light band is called as the euchromatin region. So this euchromatin region is highly significant region. Are you understanding my point? So why do I call this euchromatin region as a very highly significant region? There is a small explanation to it. You remember that when we talked about the central dogma, I told you, students, not 100% of the DNA is participating in all the functions or no function at all. Only 98% of them remain quiet. 2% of them is the one that is participating in all the functions that you see here. So those regions are the euchromatin regions. Since they are loosely packed region and they are quite active region, they're very, very active region. That is why they're very loosely packed. That is why they get their light color. And this loosely packed, the reason is very, very simple. They participate very active in passing down this genetic information. Okay, they guide your cell to prepare proteins. They create mRNA. Are you understanding? All cellular functions are controlled by this euchromatin region. Whereas coming to heterochromatin regions, they are very, very thickly packed, very dark colored DNA, which we call it as heterochromatin region. And heterochromatin region, they have hundreds of explanation. What is the role of this heterochromatin region? Why do we have so much of bulk DNA or such quantity of DNA which is not participating in much of it is present even there? Why do we even require them? So some of the scientific explanation, they say that the heterochromatin regions, they are very, very important for the structural configuration of your chromosomes, while migration of the chromosomes or while the packing of the chromosome. So it is very, very important to keep our chromosome structure quite stable. So that is the first explanation. And the second most important explanation is, so we are the organisms not just arrived like this. So it has taken millions of years of evolution for our existence. And as we evolve, we have dumped a lot of unnecessary DNA, throughout our evolution. So this evolved unwanted DNA is just packed into heterochromatin, and that is how we accumulated such volumes of unwanted DNA in our biological system. So that is what is the explanation for heterochromatin. Understood? So this is about packing of DNA in our eukaryotes. All right. So now we have a very, very important and interesting topics that are coming up. That is search for genetic material. So the journey is very, very exciting. So why it is so fascinating is because I feel that these small chapters and these small inductions that you study right now is really, really giving you some hints how your future is going to be. So if you study this search for genetic material, you is going to study about certain experiments and certain things that are done by so many scientists. So when you see, they don't directly tell what is transforming principle. Transforming principle says that dash dash dash. They don't just give you an explanation and the result of the experiment. What they teach you is the methodology of this experiment, how they conducted and what they did and what is the drawbacks, what is the results. So that is how we study this search for genetic material. So this particular topic, this is exactly even how your research happens when you go up and study your PhD or if you become, academicians. So that'd be very fascinating. So anyway, so have this interest in learning the research methodology and just try to build up this story. Okay, we did this and this is what happened and this is the results and this is how it did not conclude well. All right, let us begin with this. So the first one is the transformation principle. Now, the transformation principle or the transforming principle is famously known as the Griffith's experiment, or the Frederick Griffith is a scientist who conducted this experiment. So before we head on to this transformation experiment, I would like to talk about something called as a pseudo copulation or the pseudo sexual reproduction that happens in our monorails. So the unicellular organisms are supposed to reproduce asexually. Now you're just telling that there is some kind of sexual reproduction happening. Most of you know the answer. Some of you don't know. If you don't know, let me talk about it. So you might have heard about a process called as conjugation, right? What is conjugation? If I have two bacterias, right? If I have two bacterias, let me call one bacteria as minus and let me call one bacteria as plus. Now, what happens during unfavorable condition? Let me say that this bacteria has the capacity to live, and this bacteria has the capacity to lose a certain character or die. Now, what happens? Whatever the character this bacteria has, it is present in one of the DNA that is outside its nucleus, called as the plasmid DNA. I think you know about plasmids. If you don't know about plasmids, just put a message. Okay. So this plasmid DNA, let us say that it has a certain character. Now, whenever the unfavorable condition comes, now it will make an extra copy of this plasmid DNA, and now it will try to construct a bridge. So this is called as a conjugation bridge. Now, this plasmid DNA, which was made an additional copy, will pass from the plus bacteria to the minus bacteria. So finally, converting this minus bacteria into plus-like character or the plus bacteria. So this, just a very short explanation and a rapid explanation. So this is what is called as conjugation, right? So did you understand what is plasmid? See, somebody asked. Very good. So now this is quite nice. Let me go back and tell you what is a plasmid DNA. So where is our bacterial structure? Do we have a fine? Yeah. Can you see here? There is one circular DNA here. Yeah. So this is the nuclear region. So this is where your chromosomal DNA of your bacteria is present. So we'll neglect this. So whatever the DNA that is present within the nuclear region, that is called as chromosomal DNA. Understand? So whichever the DNA that is present outside the nuclear region, that is called as extra chromosomal DNA. Okay, I'll repeat again. The DNA that is present within the nuclear region is called as what? It is called as chromosomal DNA. The DNA which is present outside this region of your nucleus, that is called as what? It is called as what? Extra chromosomal DNA. That means it is outside the nucleus. So when I talk about extra chromosomal DNA, one such DNA that we can see in our prokaryotes is plasmid DNA. So they appear like this. If you see in the microscope, they appear like this. So what is the structure of a plasmid DNA? They are circular DNA, understand? And they are double-stranded. Double-stranded circular DNA is called as plasmid DNA. So they're very, very significant. They're very, very important for us to study these plasmid DNA. So why are we even studying that? Because whatever is present in the nucleus is what is controlling life. Definitely no. There are certain characteristics even in this plasmid DNA that enables this bacteria to survive extreme conditions. Like when you put antibiotics, if they have to adapt to these antibiotics, plasmid DNA has the genes to give them that strength. Understand? If they have to divide, for example, conjugation, I told you, if they have to make copies and send the information of their genetic nature to other cells, so that is also information is present in the plasmid DNA. That is why we study plasmid DNA very much. As we go on, we'll study how much of this plasmid DNA is important for us, for our studies also. Understood? So this is about plasmid DNA. So what is plasmid DNA? An extra chromosomal DNA. So it is very, very significant. Why? Because it has some characteristics that help the bacteria, right? So did you understand, Himamshi, right? Right. Fine. So this is about plasmid DNA, and I was talking about transformation. While I was talking about transformation, we were discussing about something called as conjugation. Very, very important. So in conjugation, what happens is A character from one bacteria gets transferred into the another bacteria through a process called as conjugation. Now, why did I call you as a type of sexual reproduction? Whenever there is an exchange of genetic information, that is almost like a sexual reproduction. So that is why what happens, there was a plasmid DNA, another copy of a plasmid DNA was made, and then they've constructed a bridge between them. And now what happens, a copy of the plasmid DNA goes from one bacteria to another bacteria, converting this minus type of bacteria into plus type of bacteria. Very simple. So this is called as conjugation. Now we understand that by this process a DNA was transferred from our one bacteria to another bacteria. But Griffith conducts a similar type of an experiment. But unfortunately, back then they didn't know about who would be responsible for this genetic information that is passed between two bacteria. Let's see the experiment. So when Griffith conducted this transformation experiment in bacteria, the organism he chose for this experiment was Streptococcus pneumonia. So Streptococcus pneumonia is an organism of interest in this experiment. So now this Streptococcus pneumonia was used against mice. So we have two organisms that are used up in this experiment. One is a mice, other one is a Streptococcus pneumonia. Now before we come to the mice, let us talk about this Streptococcus pneumonia. When we see Streptococcus pneumonia, Streptococcus is a cocci, it's a bacteria, and pneumonia causing bacteria is called a Streptococcus pneumonia. Now, if we look at the Streptococcus pneumonia, we have two variants here. That means two varieties. One variant which can cause a disease, and another variant which cannot cause a disease. So the variant which can cause a disease, that is called as virulent strain. Okay, if you tell COVID is a virulent disease, it can spread, it can cause you a disease-like condition. Any microorganism, after entering in your body, if they can cause a disease-like condition, they are called as virulent genes. Did you understand? So this bacteria which can cause this pneumonia is called as virulent bacteria. So this virulent bacteria, how can we differentiate? If you see this virulent bacteria under the microscope, their colonies or their group will have a very smooth coat around them. So that is called as mucopolysaccharide. So this mucopolysaccharide will give them the smooth appearance on their external surface. So that is what makes the colonies of this bacteria look very smooth. So the smooth bacteria, smooth looking bacteria. What is smooth looking bacteria? If I look them under the microscope, they look like very gelatinous, very slippery. So those are called as smooth strain bacteria or the virulent strain of Streptococcus pneumonia. And the other strain or the other type of the bacteria that he works was on the rough strain bacteria. So their colonies look really rough because of the absence of this mucopolysaccharide. And when you look at their colonies under the microscope, their colonies look quite rough. Okay, like rocky. So they are called as rough strain of bacteria. Understood? Now, when you take the smooth strain of bacteria and inject into the mice, it will cause pneumonia, obviously. So if I take one pneumonia causing bacteria which has virulency and give it in your body, what happens? You get a disease, obvious. Whereas the R strain bacteria, if I just put it in the, in the organism, then it will not be able to cause any disease. So that is why it is called as non-pathogenic. So smooth strain is pathogenic and the rough strain is non-pathogenic. Now what happens? Griffith's experiment happens like this. He takes first experiment, what he does is, he takes the smooth strain and he injects into the mice. So what happens? The mice gets pneumonia. After pneumonia, the mice dies. Simple. Next, he takes the rough strain and then he injects into the mice. The mice will not get the pneumonia because it is non-virulent, and then the mice will live. It is the two basic things that we just discussed. Next, what he does is, he takes the S strain and then he heat kills it. Now, many people will misunderstand this term. Heat killing it. That means not boiling and killing the entire bacteria. No, we are not, boiling it, not boiling the bacteria. What happens is, whenever we do a certain process in microbiology, we call this process called as attenuation. Okay, we call this process called as attenuation. So what is this attenuation? Whenever, if you have a virulent bacteria or the disease causing bacteria, and suddenly when they are increased to higher temperatures, Okay, when they're increased to higher temperature, higher means around 40 degrees Celsius. Okay, whenever the temperature is increased to 40 degrees Celsius, and this shock of this heat will make them lose the character of virulency. That means whenever we just take the smooth strain bacteria, we put it in a beaker, and when we suddenly increase the temperature to 40 degrees and we take that out from that heat, now what happens is this heat shock will make them lose the genes that are or the DNA that is responsible for them to cause the disease. Okay, how it happens? You know that ultimately, if you have a gene for a particular character, let's say that virulency or disease causing is a character. So when they increase the heat, obviously this gene will give RNA and RNA will give rise to proteins, right? So when the heat was increased, now what happens? The proteins that were supporting this bacteria to cause a disease got denatured or destroyed because of the heat. Now that made this smooth strain bacteria non-virulent. So what happened in the third experiment? He takes the smooth strain, he heat kills it. That means he suddenly increases the temperature, and then he takes that bacteria and then injects into the mice. The mice will live. What is the explanation for this? When he increased the temperature, now we know scientific analysis of it. When we increase the temperature, the proteins that was helping this bacteria to have this virulency got destroyed. So that is what made this bacteria non-virulent. And when it was injected into the mice did not get affected. It started living. Now what he did? He takes this same heat-killed bacteria and then he mixes with the rough strain. Are you understanding? So let's take, here it is, sorry. He takes this heat-killed smooth strain and he mixes with its rough strain. Now what happens? The character from the virulent character from the smooth strain got transferred into the rough strain. Now when he injected into the mice dies. Now what happened? Griffith got shocked. There was something that was transferred between the smooth strain to the rough strain. That is what made this rough strain bacteria to become virulent and kill the mice. Now, what was transferred between the smooth strain and the rough strain, he didn't know. But I will tell you the explanation now. What happened in this experiment? When he mixed this smooth strain and the rough strain heat killed smooth strain and the rough strain bacteria in the third experiment, what happened is, now we know that the proteins that are helping this smooth strain bacteria is destroyed when he heat killed it, but the DNA is present. Now, when they mixed it both, due to conjugation, the genes that is responsible for this virulency get transferred from smooth strain to the rough strain. Did you understand? But the rough strain bacteria already had the proper proteins or very good proteins, healthy proteins that can cause this disease. Understood? So since they had the proteins and only the between the smooth to rough, so now that is what converted this rough strain to the smooth strain or the virulent strain, and that is what killed the mice. But unfortunately, Griffith could not have the biochemical analysis to until what made this smooth strain to convert the rough strain into the virulent strain. He could not explain. Correct? So that was the problem with respect to Griffith's experiment. Understood? So in this experiment, what is the conclusion? The rough strain bacteria was transferred by the S strain of bacteria, and the transformation of the R strain to the S strain is due to the transfer of genetic material, but you could not biochemically tell whether it is protein, whether it is RNA, whether it is DNA that was transferred between these two. Understood? So can we revise this transformation? Very simple. What is happening here? Conjugation is happening. What is the organism? Streptococcus pneumonia and the mice. What are the two strains? One is the rough strain, other one is the smooth strain. Rough strain is non-virulent, smooth strain is virulent. Now you take the smooth strain, inject into the mice, it is virulent, it gets pneumonia, the mice dies. You take the rough strain, you inject into the mice, nothing happens, it is non-virulent, the mice lives. Next, what you do, you heat kill it. When you heat kill the smooth strain, the proteins in the smooth strain get destroyed, which can cause an infection, but the DNA is fine. So when the heat kill smooth strain was injected into the mice, since there were no proteins to cause a disease, now what will happen? The mice will live. Now you take the heat kill smooth strain, mix it with the rough strain. Now I told you about conjugation, the character of the smooth strain transferred into the rough strain. Already rough strain had these healthy proteins or the disease causing proteins. So now that converted the rough strain into the smooth strain, killing the mice. So ultimately, Griffith could understand that there was something genetic material was transferred between S strain and the R strain, and he could not conclude what is the biochemical nature of this genetic material. So that left another gap for the people to research upon it and analyze what might be the genetic material. All right. So time's up. If you have any doubts, students, please let me know. And you have 2 minutes to ask the questions. And as usual, I'll see you in the tomorrow's class, and we'll just revise and continue with the rest of the chapter. This chapter is quite important, so that's why I'm emphasizing on the very technical things and repeating as many times as possible, because when you read it, some of you might get confused and some of you might feel it's too difficult to understand or remember. It's very easy chapter, okay? It can be on your fingertips. So let's try to achieve that. So thank you for your time. I'll see you in the next class. If you have any doubts, here you go. You can just drop it in the message. See you.